All right. I think I would then like to call the meeting of the Scientific Advisory Committee Subcommittee to order. My name is Kathleen Corrado. I am the chair of this subcommittee, and I'd like to welcome everyone for being here today, and especially thank the subcommittee members for taking the time out of their schedules to be available for this meeting today. Before we get started, I would like to remind all the subcommittee members that we need to have your video camera on at all times during the meeting. I would also like to remind everyone to please speak up to make sure that everyone can be heard clearly. And I would also ask if members could use the raise hand function if they have a question so that we can make sure that we're recognizing that you have a question. Before we get started, I'm going to ask the members of the committee to introduce themselves. Basically, just tell us what part of what your role in the Scientific Advisory Committee is. I'll start first. My name is Kathleen Corrado. I'm chair of the Scientific Advisory Committee, also chair of the subcommittee, and my role on the advisory committee is as a American Society of Crime Laboratory Directors member. I'm then going to call on the next members. I guess I'll start with Dr. Vallone.
Sure. My name is Peter Vallone. I work for the National Institute of Standards and Technology. I'm the group leader of the Applied Genetics Group. My background is in DNA forensics, reference materials, and Kathy, I act as the co-chair to Kathy for the committee. Thank you.
Thank you. Ms. Forey?
Good morning. My name is Erin Forey. I work for RTI International in the Center of Forensic Science Advancement and Application, and I represent quality assurance and quality management on the SAC.
Thank you. Ms. Bro?
Good morning. My name is Jennifer Bro. I currently sit as the director of Montgomery County Police Crime Laboratory. I came from the forensic biology side of the house for many years. So I sit as a representative on the forensic biology side, and I am brand new to the committee this year.
Great, welcome. And Dr. Maha?
I'm George Maha. On September 30th, I retired from LabCorp, so I'm comfortable at home. At LabCorp, I was an associate vice president, and I was the lab director over the DNA identification, histocompatibility, and consumer genetics sections. And I serve as a population geneticist and biologist, DNA biologist on the committee.
Thank you, and congratulations on that retirement. Well deserved, definitely.
Thank you. It's been a long running, 36 years.
That's crazy. Thank you all for the introductions. I also would like to notify members of the public that we will have the opportunity for comments to be taken at the end of the meeting. And as was indicated on the agenda of the meeting, public comment will be limited to 5 minutes per speaker. The first order of business that we have today would be adoption of the agenda. I think the subcommittee members should have received a copy of the draft agenda from Amy Jenkins. I would ask if anyone has any changes, and if not, if we could have a motion to adopt the draft agenda.
So moved.
Second.
Thank you. So the agenda has been adopted. I'm sorry, I need to take a vote on that. All in favor, committee members in favor of adopting the agenda, say aye.
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 Any opposed? Hearing none, the motion passes and the agenda is adopted. The next part of the agenda will be new business, and we have 2 presentations for us. The first will be an overview of the podcast so the committee members can become familiar with the podcast, and the second will be an overview of the serology and historical procedures of the department. So first, I'd like to invite Department Counsel Amy Jenkins to provide our first information regarding the podcast overview.
Thank you, Dr. Corrado. Let me go ahead and share my screen. Are you all able to see the PowerPoint?
Yes.
Thank you.
Yes.
So Dr. Corrado asked, as we were preparing for this meeting, that I provide kind of an overview of the department's knowledge of the admissible shreds of evidence podcast, and also provide what documents had been received and a final or some kind of understanding as to what the status of those cases were that had been provided by Virginia Public Media. First off, I thought I would go through the department's knowledge of the podcast and when we first became aware. Let's see if I can advance this. It's not advancing for me. Let me go here and see if it,
Make sure you've clicked on the, there you go.
there we go. Is it advancing?
Yes.
Okay. So DFS was originally contacted by the podcast reporters in 2019. They began by requesting records under the Virginia FOIA Act. The first set of documents that were requested were personnel files, and then additional FOIA requests were received all the way through February of 2022 to include some case files where they provided us documents in response ultimately on those case files. The podcast reporters were interviewed by Director Jackson and Brad Jenkins in February of 2022. And at that time, DFS became aware of the documents referenced in the podcast and those were referenced in the interview. And at that point in time, DFS requested copies of the documents from the podcast host subsequent to the interview. We received a response from VPM thereafter in March of 2022. They declined to provide copies or access to the documents at that time. They indicated that they would reach out to DFS once they were further along in their investigation. And at that point in time, we had no more contact until 2023. As far as podcast reviews, I would encourage you all to take a look at the transcripts for the podcast. I found them to be very helpful, and if you need assistance in locating those on what I sent you, but there is a transcript for each of the episodes posted. The podcast focused mostly on Mary Jane Burton. That name may be familiar to all of you. She was a serologist who worked at the Bureau and then the Division of Forensic Science from 1973 to 1988. She had, for a portion of her time here under employment, retained clippings of evidence in the files that she worked, taped down to her serology worksheets. As was noted in the podcast, she was not the only serologist who did so at that time. The first episode of the podcast focused on the exoneration of Marvin Anderson. Mr. Anderson's case was the first case where an individual was exonerated based on the DNA testing of those taped down swabs or clippings in the case file. Those swabs and clippings had been discovered at that time by Paul Farrar, who is the previous director of the Department of Forensic Science. The second episode consisted mostly of interviews with Gina Demas, a former serology trainee with the Bureau, and Shirley Patterson, a former secretary. There were allegations that Burton testified at trial, and that was observed by Ms. Demas, that a hair had originated from the defendant and no other. Ms. Demas indicates when confronted, Ms. Burton indicated that the Commonwealth should not be trying the man if he was not guilty, and explaining why she testified in that way. Additional allegations include that Burton did not run controls or take the temperatures of gels, as she would just run it slowly down the test tube. She would also make calls for murky electrophoresis results. It's also indicated that Ms. Burton accused the secretary, Ms. Patterson, of mistyping a result in a report of the suspect's blood, and subsequently was observed shredding the report. Ms. Patterson indicated she did not mistype the report, but Burton had her retype the report and mail it out. At that point in time, staff members began photocopying bench notes looking for discrepancies in Ms. Burton's file. And Ms. Demas and Ms. Patterson photocopied lab record books before and after, indicating that Burton changed results with no additional testing. Ms. Demas filed a complaint to Warren Johnson, then head of the lab, and to Paul Farrar, who at that time was the chief chemist. The third episode consists of the podcast reporters obtaining a box of documents from a lawyer in Richmond who they indicated was connected to the DNA post-conviction testing project. They described the box as filled with documents. They reviewed the documents, although it's not really clear how many, with Marilyn Miller, a retired VCU professor. The focus was really on a 1977 rape case where it was indicated that item 33 in that rape case was changed to reflect the suspect's blood type, EAP S-race D, indicating that it is a case or was a case that Burton shredded her initial report in. And then they also discuss Burton's use of pencil for her notes and her failure to conduct species testing, specifically referencing what came to be known as the cat case and a hunting accident case. Ms. Joan Fonts, a previous serologist with the Bureau, was contacted by the podcasters also in this episode because they were told that she supported all of the concerns that had been raised regarding Mary Jane Burton. Ms. Fonts does not speak with the podcasters on the episode. The fourth episode is really focused on background on Ms. Burton and the fact that she died in 1999. The fifth episode begins with the reports made to Warren Johnson and Paul Farrar and discussions on various meetings that were said to have occurred that where Burton's errors were discussed. The podcast then switches to a discussion of the Julius Ruffin exoneration and Burton's testimony in that case, where she indicates the perpetrator was a type B secretor and about 8% of the people are type B secretor. The prosecutors then asked her what secretion type Ruffin was. She indicated that he was a type B secretor. There were no allegations or documents provided in the podcast indicating that the results in Mr. Ruffin's case, serology results, have been changed. The podcast then notes that Ms. Burton began working on weekends. There is an allegation that she asked Ms. Demas to forge a chain of custody form, to sign a chain of custody form, and that when Ms. Demas refused, Ms. Burton forged Ms. Demas's name on it. At that point in time, Ms. Demas files 2 whistleblower lawsuits in the U.S. District Court for the Eastern District of Virginia. She indicates that she and other serology employees, including Ms. Fonts, Mr. Elmer Gist, Deanne Dabbs, and June Brown, go through hundreds of cases to find case after case run by Ms. Burton without controls. The first lawsuit is dismissed for a discovery violation, according to Ms. Demas. The second lawsuit proceeds to discovery, where Ms. Demas indicates she was deposed for 2 days by a deputy of Marshall Coleman, who was then the Attorney General of Virginia. The deposition lasted for 2 days, and that she provided several stacks of documents to Marshall Coleman as part of that deposition. The second case was dismissed for what lawyers would know as res judicata, meaning it wasn't substantially different from the first lawsuit and had already been determined. So it was dismissed by the court. The podcast then indicates that Ms. Demas was moved out of the serology laboratory. She indicated that the box of documents was taken out of the laboratory by another employee, and that she subsequently resigned after a year, filing one additional complaint with Dr. Tiedemann, who at that time was the head of the Virginia Department of Health, and the department was still under the Department of Health at that point in time. We were not our own department. The sixth episode involves interviews with Deanne Dabbs and Pete Marone. Dabbs indicated that Burton had no written protocols, no inspections were conducted of the laboratory, and there were no quality control measures. Her notes were not complete. She shared concerns and felt that Burton should not have been allowed to continue working, or an in-depth look at what she was doing should have been appropriate. Then Pete Marone, the former director of the laboratory, was interviewed by the podcasters. He indicated that he was hired to improve the procedures at the serology laboratory. He indicated that the issue was sloppy note-taking, and that his review of the cases indicated that the cases had incomplete notes and the clippings were not properly stored, or were not the proper storage of evidence, and the practice of retaining those clippings in the file was subsequently stopped. He indicated that notes should be taken in pen and not in pencil. And subsequent to that, it's noted that Burton retires from the Bureau, or from the division at that time. The lab subsequently sought accreditation, and Dabbs indicates in a follow-up interview that having been shown what the change of results of item 33 from the rape case, that it was bad documentation, and if the results were changed, Burton should have documented why the results were changed. The 7th episode focuses on the 3 exonerations after Marvin Anderson. It also talks about that subsequently Governor Warner ordered that 10% of the takedown evidence be tested by the department. 31 additional cases were identified that were suitable for DNA testing, and 3 additional exonerations resulted from that testing of those additional 31 cases. Governor Warner then orders that all cases with clippings viable for DNA testing be re-examined, and 7 more exonerations occur during the PC DNA testing project over the course of a number of years. The episode highlights complaints about the process for the PC DNA testing notification project and the testing, and the notifications to defendants by the Forensic Science Board, DFS, and the Crime Commission of the potential for DNA to be in files that was amenable for testing. The 8th episode focuses in on several of the additional exonerations: Winston Scott, Willie Davidson, Victor Burnett, Thomas Hainsworth, and Bennett Barber. There were additional allegations against Burton on each of those exonerations. For Scott, the podcasters indicated that Burton's first round of testing excluded Scott. Burton reruns the test and reports both the perpetrator and Scott are type O secretors, and she attributes her changes at trial to bacteria on the genes affecting the serology typing. On Willie Davidson, it's indicated that Burton overstated the serology results at trial, indicating that 42% of the population would have been excluded when no one would have been excluded from the results. Victor Burnett, there's also an indication that Burton provided misleading testimony. Thomas Hainsworth, Mr. Hainsworth was told leading up to trial that his blood type was a B, but at trial it was stated that he was a type O. And Bennett Barber, it's indicated that a blood test could have been run that might have excluded him depending on the result. The 9th episode focuses on 2 additional exonerations, Julius Ruffin and Arthur Whitfield. Both men were convicted based on victim identification, and the cases are discussed at length. Subsequent DNA testing showed that Aaron Doxey, unknown serial rapist, was the perpetrator. They also interviewed one victim in the Whitfield exoneration that believes that her genes and the other victim's genes were swapped during testing. Both rape cases occurred on the same night and were worked at the same time by Mary Jane Burt. The 10th episode focuses on a discussion of whistleblowers. The 11th episode begins with the interview of Director Jackson. It goes into criticism regarding the PC testing project, arguing that it did not get to the root of the problem, which was the underlying serology testing. Earl Washington's exoneration is discussed, and the ensuing audits and reviews. The initial serology testing in Mr. Washington's case indicated that the perpetrator had transferrin CD, that marker, but after a meeting with law enforcement, the results were changed to inconclusive. The serologist in that case was Deanne Dabbs. In a subsequent deposition, Ms. Dabbs indicated that she had stumbled on articles regarding transferrin that made her question the results. The podcast reporter indicated that he could not find those articles. It then talks about how the governor at the time ordered an independent audit and Washington was granted a full pardon. The Keith Harward exoneration is also discussed, where the original serologist called it inconclusive, or an inclusion rather, but it was an exclusion, and those exculpatory results were not reported. The serology review is discussed in that episode, where approximately 200 cases were reviewed by the Scientific Advisory Committee and DFS staff. No other instances of exculpatory results were reported, and the Innocence Project offers its critique of that review, indicating that it was not transparent, but acknowledged that the allegations of changed results in a logbook, as is alleged in the current podcast, would not have been found during the course of the serology review. The 12th episode focuses on forensic laboratories being too enmeshed with law enforcement and examiners being too susceptible to influence and implicit bias. So that's a review of the overall allegations in the podcast. And again, I would encourage you to look at the transcripts, because they make it a little bit easier to follow who's speaking. After the podcast aired, DFS didn't learn that the podcast had been published until February of 2023. It began in January of 2023. In May of 2023, the podcast reporter asked for an on-the-record interview in exchange for providing the requested documents. DFS declined the interview request, but renewed a request for the documents that had been made back in February of 2022. In July of 2023, VPM provided the documents that you have received for your review, and they have been posted on the website in a redacted format. Just so you all are aware, the records that we do not have a personnel file for Mary Jane Burton. We don't have any documents pertaining to any complaints made concerning Ms. Burton to anyone at the Forensic Science Bureau, the Division of Consolidated Laboratory Services, or the Virginia Department of Health. We don't have any records from the lawsuits filed by Ms. Demas, only the newspaper articles. And the Central Laboratory Serology Logbook referenced in the podcast is also, we haven't been able to locate it, or has been destroyed. This isn't inconsistent with the Commonwealth of Virginia's record retention destruction policies, as the timeframes set for the record retention and destruction of all of those different documents has run. As to the documents provided by VPM, Dr. Corrado asked that I provide some kind of overview of what we have and where these cases are, what the status of these cases are at that time, or at this time rather. 75-10986, which is referenced in the podcast as the cat case. The allegation there was that there was no species typing that resulted in the loss of the case. There were 3 co-defendants that stood trial for that case. None of them were subsequently convicted. It's a pretty long and drawn out legal tale, but basically 2 of them were convicted. It was appealed to the Supreme Court of Virginia. The Supreme Court of Virginia reversed the convictions, indicating that there had been an evidentiary failure by the court pertaining to an item of evidence that tied one of those co-defendants to the homicide. It was sent back for retrial, and on retrial, there was the initial jury found, or subsequent jury found not the individual not guilty. The second individual, the case was null prost against them. And all of this was kind of reviewed as part of the PC project. And this case was deemed ineligible because there were no convictions as a result of the case. There is a letter in the case file, and the case file has also been posted from Pat Bynum, who was the prosecutor of this case, responding to allegations that Ms. Burton was the cause of the loss of the case, that I would bring to the subcommittee's attention. The second case is the rape case, and this is the one that I believe is the item 33, the change of the suspect's blood type, and other discrepancies were alleged in this. This also was ineligible for the PC DNA testing because the charges against the two co-defendants were reduced to fornication with a fine imposed. So as misdemeanor convictions, it was not eligible. We were not able to locate one of the co-defendants, and the other co-defendant was deceased in this case as far as the PC notification side. The third case, the Fairfax murder case, the allegations there are discrepancies, errors, and alterations of test results. And in this case, testing was done as part of the PC project. They did determine that the victim's blood was located on the suspect's pants, and the inclusion was 1 in 1.1 million, and I know I'm probably not doing that with the scientific accuracy that it deserves, but the scientists can clean me up later. They were unable to locate the defendant on this one, so there was no notification that we were able to make. 769468 is referenced as the hunting accident case or the hunting case. The allegation here was that there was no species typing done when requested. This was one that was not in the PC case database at all, likely because there was not any taped down evidence in this particular case. I have confirmed with the law enforcement officers, not the actual original detectives, but the agency themselves, that they don't have any additional files on this case and that no convictions resulted. And then 76-23383 is an inmate felonious assault. The allegations here are that blood stains on the suspect's pants were reported as being consistent with the victim's typing when testing indicated that they were consistent with the suspect's typing. This was another case that was deemed ineligible by the PC project because there's no record of anyone's conviction on this case. There is a sixth file that I have provided to the subcommittee members that was requested by VPM, but we did not get any documents from VPM in regards to it. That is 74-13449. That's also been posted, redacted on the website. And that dealt with item 33 as well. And the defendant was not eliminated in that post-conviction testing for that. And the defendant was deceased. And there's a letter from the prosecutor in the case file for that regarding the DNA results that were reported subsequently and indicating that the defendant had died in the Department of Corrections. So that is, in essence, my overview of all of those things. And I'm going to try to stop sharing now.
Thank you, Ms. Jenkins. That was very comprehensive, very helpful, at least for me. I'm sure it probably was for the other committee members as well. I think at this point, we'll move on to hear from Mr. Scanlon to give us a serology and procedure overview from what the lab was doing at the time. And then after that, we will have the subcommittee. We can open it up for some questions and discussion. So, Mr. Scanlon.
Everybody see that okay? And hear me okay?
Yes.
We'll turn it over to Bob.
All right. Hi, I'm Bob Scanlon. I worked at the for the division or the department and the bureau. My tenure goes back to 82 1982, but I was forensic biologist for a couple of years prior to that. So I came here with experience. I actually did work under Mary Jane Burton, but I was not trained by her because I had already been court qualified and was, was testing evidence and testifying at that point. So what I just wanted to do was do a very brief walkthrough about some of the things that we were doing back then. You've got to kind of walk back in time a little bit. Virginia Crime Lab system started in the early 70s. It was not accredited until 1989. And at that point, we had already begun doing some DNA testing. But it was conventional serology, strictly conventional serology during those early years. So we're talking ABO testing. We did RH, some RH testing. We were doing polymorphic enzymes, serum proteins, and some blood serum proteins were also included in that for blood samples. So in 1994, of course, that was discontinued, replaced with DNA testing. And, at that point in time, lots of things, of course, changed. I wanted to kind of set your mindset a little bit back in time, because this really was the infancy, early days of forensic biology testing. Back in those days, and when I arrived in 82, there was no specific procedures manual. There were some brief lab write-ups for different procedures that one could follow, kind of like you would be following in a college laboratory course, step 1, step 2, step 3. But there was no procedure manual written. Lab accreditations were not happening yet, so that predates any quality assurance documents. External proficiency testing programs were not yet on board. Method validation occurred by, there was serious science put into it. Of course, people would go to a meeting, learn about a methodology, bring it back to their laboratory, test it out on known samples till they were satisfied that it was working well. And, I'm sure there was some management involvement in allowing it to be put online, but that was the extent of validation. The examiners, one thing that was interesting and actually somewhat helpful at the time is that examiners received the evidence directly from the investigators. I say that's a good thing in that you had that couple of minutes of direct conversation with the investigator of the case to ask him, hey, I don't have this known sample, or I need this, or what is this? What's your most important piece of evidence? Where did this piece of evidence come from? Or maybe tell them you didn't preserve this very well. Here's how you should do it in the future. So there was some value to that. There were not computers, for reports. They were dictated into a dictaphone, which I never got the hang of. I would handwrite mine and then give it to the secretary, but they were typed on a typewriter. So you can imagine that allows for, some clerical errors and things. So you have to be careful with that. And there was, as far as peer review of analytical results, it did take place when I arrived in 82. We were all reviewing each other's reports. I'm not sure if that was 100% requirement before that, but when I arrived, Pete Marone had already been here and had begun to start implementing best practices in the laboratory and sort of revising some of the practices that were going on as far as documenting of results and what have you. So I just wanted to kind of throw that out there because, it's difficult to look back at some of these case files and you say, oh, you just kind of hit your head like, my goodness, this would never fly today. But at that time, there was a lot of evolution to occur in forensic biology labs. And that did take place over time. I want to make a comment about bench notes. Today, I think quite often when case goes to court, those bench notes are brought in for discovery. It's almost like an automatic thing these days. At that time, that was almost unheard of. Purpose of bench notes were viewed as being for the examiner's use, and so that you could have sufficient information to recall what you did and write a report. They were not nearly as complete then as is currently done. However, again, best practices, some of the scientists, I know I like to take more voluminous notes, put down more observations, but not everyone did that. And Mary Jane Burton's notes were very, very skimpy. She didn't put a lot of detail into them. Just something that I observed. But there were some optional worksheets that we were using to record data. And I wanted to just show you a couple of quick things. You'll see them if you are able to look at the case files, but I wanted you to kind of know what you were looking at. This is an example of one of the worksheets, but sometimes it was used for both secretion typing as well as for blood sample typing. And you actually can see down on the bottom there some of those swabs and cuttings that would be used in testing, and then Mary Jane Burton would tape them down into the case file after she had done the testing on them and leave them there. Again, when I was here, that practice was being stopped, and I never did that. I know a lot of the examiners, frowned upon that, not the least of which it was just kind of unsanitary to have this case file on your desk with biological material on it. Let's take a quick closer look here at some of the information on there. So, for blood identification, if you can believe it, it was still the benzidine test, which is now, benzidine is a known carcinogen and no lab uses that anymore, but it was a test for blood, one of the first, and it was used then, as well as phenolphthalein, which is still used. And then there was the precipitin test, was for species testing, was either going to be done by double diffusion on the so-called Ochterlony double diffusion gel plates, or in a very narrow, thin tube where you would put the antisera in and then take the extract and layer that on top and look for a precipitation reaction, saying, recognition for species testing. Run that along with controls, of course. Further down, you'll see a lot of these worksheets in the case files. So for antigen testing, we were actually doing ABO testing as well as RH testing, absorption elution, which was a direct antigen test, was being done. So what I have circled there in red is, you would have the A, B, and H reaction, and then you look at the result over there and you see that, you get a positive for the A and the H, which indicates type A blood, and no reaction for the B. I can't help but notice things when I'm looking at that, like, instead of putting a 0 with a line through it for no reaction, you put a dash, and you could easily put another vertical line through it and it turns into a plus. That would be a best practice that I would want to do. But again, this is what she did. And many examiners, when they were grading the reaction, it would be no reaction, +1, +2, +3, or +4, +4 being the strongest reaction. most often in Mary Jane's files, it was either plus or minus. It was all or none. But you know, this is what she recorded. You'll notice to the right of the red circles, there's the blue lines. So this is the recording for RH factor. So capital C, capital D, capital E, little c, little e are the RH antigens. If you possess the big D, you are RH positive. If you do not possess the big D, you are RH negative. And I know Mary Jane liked this test. She did it a lot. Later on, this test was dropped. It's a much weaker reaction, harder to read and less reliable. So that was dropped later, but it was possible to do it by absorption elution. So again, you could see her reactions there, minuses or pluses in the same column, but on the right side. That's what that's referring to. And then also you'll note on there that she would put her HB would be haptoglobin, serum protein, down the bottom, EAP and esterase D, those types. So we were talking about, I don't think I photographed the logbook, but because they were pretty bad copies. But these would be the enzyme typing results that would be summarized on here. There was a logbook. Yeah, there was a logbook where for enzyme typing, you would have each of the lanes numbered for an electrophoresis run, and then the sample names and the typing results that you got. Very often what you'll see in those files is just one reading of that, those results. Later on, what was implemented is that there would be a first read and then a second independent read by a second examiner. And then you would compare your results. And if you agreed, then, then it would be called. If you didn't, then it was not called. Most of the ones you're going to see in here in Mary Jane's are just the one read. I think because of the first read, second read, and then recording or actually taking a photocopy of the logbook and putting it in the files later on, that was the reason why those logbooks were not retained. They got rid of those. So, but you'll see the summary things in there, and you'll see some of those results that I think Gina Demas and Joan Fonts had made photocopies, as well as Ms. Patterson, the secretary, was making some photocopies of some of the case file notes, and you will observe some of those in the data that's being provided. Yeah, and even sometimes, you'd see results for like a known sample or something like that recorded directly on the lab request form, which is just not really a good practice, but it happened in those days. Oh, and this is so with secretion typing for seminal fluid, saliva, those types of samples, you'll see some of the absorption inhibition secretion typing. If you're not familiar with that, it involves taking diluted so-called titered antisera that is just strong enough to react with indicator cells, and then placing the sample A, B, and H pieces of material in there. And if the type exists, it'll further absorb the antisera, thus preventing it from reacting. So it's like a negative reaction. So you'll see, in this one, A, B, and H. So the conclusions would be type A because A and the H sample are inhibited. They do not react, but the B one does. So proof positive that it's present is the negative reaction, if you will. And you notice that there is a control sample next to that. Also, amylase was run to detect saliva but very often that was not reported. Amylase was never really viewed as a very reliable test, but it would give you some indication that saliva could be present. So sometimes you would see that, but it's not reported in the results. That was not unusual. Just a couple of other general observations. In those days, there were no written conclusions comparing the results to the reference samples. That didn't happen until the 80s. We would explain them in court or, to the investigators if they wanted to know. No statistics were included in those reports, although you might find some calculations in the notes, especially if someone went to court, you would talk about statistics then. And at times, if there was nothing to compare a known reference sample to, then you might find that there were no results reported, but they might be in the notes, and vice versa. If there was no reference sample, perhaps the samples, the evidence samples that were run might not be reported at that time until you had something to compare it to. It was in those days, it was not unusual to have to really go back and beg for for known samples to be submitted to make comparisons. There were some body fluid screening results recorded in the notes, but not reported. As I mentioned, amylase was an indicator of saliva, but very often not reported. So it's just some things that I wanted to point out. You'll see things as you go through, but I kind of wanted to give you a little bit of a guided tour.
Thank you so much, Mr. Scanlon. Again, this is very helpful. I received a lot of this information and started looking through it, and I think a lot of my questions that I came up with, you already addressed. So thank you for that. It's helpful. At this point in time, I would like to open the agenda up to any members of the subcommittee that have any questions. I would say that I do have a short list of questions that I would like to ask the DFS, but mostly, not that I necessarily expect an answer, but questions that I was sort of coming to me as I was starting to review things. And, if maybe we can, if there's any questions we have or any additional information that we think would help us in our review, we could bring that up now. So I will ask, I think I'll ask the other subcommittee members first if they have any thoughts or questions that they want to bring up at this time. Again, I just want to make it clear that this is a lot of information that we all received, and it's very time consuming to go through it. So I don't think we would have expected anyone to go through all the information yet. But this is just an opportunity for us to ask some questions, and then so we can review it in more detail, and we likely will have to have another meeting set up after this. So do any subcommittee members have any questions or thoughts they want to bring up at this time?
I have some thoughts. And for the record, this is George Maha. Being retired, I had time to listen to the entire podcast. So I have reviewed the podcast itself, and I thank Mr. Scanlon. That was a nice overview. I would have probably said some of the same things. I also go back to serology days. So although I did it on the clinical side, but I am familiar with the typing process and HP and some of those other red cell enzymes, which I personally hated at that time, but was very happy to see DNA come along.
Yeah.
I think there were, in the podcast itself, Amy caught one of the things I would have brought up was that what these reporters were looking for was for this thorough review, except that the thorough review wouldn't have revealed anything unless someone had photocopied the original logbooks And then we had the altered ones. And so that review, that kind of review she seemed to be looking for was not realistic. And that you were just not going to find anything. I mean, you might, I think Brad found a typo in the thing we did a couple years ago when they did the sampling review of some of the serology cases, but there was nothing that looked like there was anything nefarious going on in any of those cases they reviewed. The other thing they kept talking about was what would happen. Did you look to see if there was a difference in the results compared to what you had with DNA, which I guess was not done in the comprehensive retyping back when Governor Warner had ordered. And the problem with that is sort of one of the things we talked about in there, is Amy had brought up, there was a marker that was found in 8% of the population, which is about 1 in 12 people. So if you retype with DNA by today's standards, of course you're going to find some differences. That's exactly what you would expect. But it's not because someone did something wrong in the 1970s. It's the limitations of the technology that was available in the 1970s. And I mean, you did the best you could with what you had. And you know, by today's standards, no one in their right mind would use serology. It's too primitive. So I think those are just sort of two comments I got from what these reporters were looking for. And in neither case would they be what they want would be useful, or what you wouldn't find anything unexpected. And in fact, you could argue the same thing if you went back and redid cases you did with RFLP in the late 80s and early 90s and did them with today's super STR panels we have, you would find discrepancies too because of the limitation of statistics. It becomes much less likely. If you did 4 RFLPs, it might be 1 in 200 or 1 in 300. You would have some really small number, and most of what you'd pick up would probably be cases where a sibling was the actual perpetrator rather than the person you had. It would be a relative involved, would be my bet. But a random person would again be very rare, but would happen. So that what they wanted, I think, was not realistic, and or not useful, might be a better way to put it.
Thank you, Dr. Maha. That's very helpful as well. Does anyone else have anything they want to bring up? I don't see any hands. Someone correct me if you do. I have a few questions I guess I would like to ask if that's okay. I think I'm probably referring these to Mr. Scanlon. And again, you may or may not be able to answer them at this time, and that's fine. I guess I'll just start with my list. My one question I had was, I noticed in some of the case files that I was able to look at that it seemed to be multiple copies of the request for lab examination document. And I think you mentioned this in your presentation. Sometimes there would be handwritten notes on them. Sometimes there wouldn't. I mean, do we know what was the practice at the time regarding those? Why were there so many copies in the case files? So that's one question.
Well, sometimes, there would be one master copy, I guess, once all the recorded chain signatures had gone back and forth. But there could be various and sundry, midpoint requests for lab exam that, had like the initial receipt, but not the return of the evidence, for example. So it's very common to have multiple copies of those in the case file. Essentially what you would want to do is look for the most complete one, and that would probably include the return of the evidence to the investigator after the testing was done. But that's not an unusual thing.
Okay, that makes sense. Thank you. You also mentioned in your presentation that at the time, a lot of the reports were dictated and then typed. or perhaps handwritten and then typed. Do we know if the practice of the lab was to retain any of those drafts, or was it once the report was typed, then the drafts were no longer needed?
I'm pretty sure the dictaphone recordings were not saved once the report was released. I think sometimes there would be handwritten stuff in there, but I doubt seriously that you're going to find those in any of Mary Jane Burton's. She was a big proponent of the dictaphone method. Some of the later examiners, like myself, preferred to just handwrite a report.
Yeah, I was like you.
Yeah. And so the handwritten ones might be in there, but I
Okay.
don't think you're going to find any in hers.
Okay. And as far as the case files that we received copies of from the department, and I don't know if anyone could really answer this, but my question is, I think, do we know how complete those case files are? In other words, to the best of our knowledge, do we think that all of the documents that belong to that case were in that case file? Or is it possible, for instance, if things were batched, if multiple cases were batched, that there might be a worksheet that's going to be found in another file and not in the file that we think it's in? Or I guess anything along those lines. I mean, is it possible that there's information that's missing that we might not be aware of?
I think it should all be in there. However, I started looking through some of these case files already. I haven't made it the whole way through all of them, but I've looked through several of the ones that Amy mentioned. And I did note that in the official case file, she provided me with the VPN, the documents that were provided to us, and then separate from that, the case files. And in looking at the official case file that we had, some of those logbook photocopies were not in the official case file, but they were in what VPN had, because I think that's what my presumption is. That's what Gina Demas was saving and preserving for, as proof or whatever later of changing things. And actually, to Dr. Maha's comments about, it would be helpful to have a before and after. There are some of those. There are some worksheets, logbook copies in that are labeled before, and then some that are labeled after, that were in at least one or two of those case files. But these, so far as I've gotten, those were the ones that were provided by VPN. So, not to be overly, concerned about some conspiracy theory, but that which we received from Gina Demas and from VPN, isn't our official one. So we have to be very careful about relying on that. Did Ms. Demas have did she make some alterations or something like that? We don't know. So I think we have to be very careful to rely on what is the official record that we have. That being said, there is information in those documents that were provided by VPN. I've seen some things that both Gina Demas and Joan Fons had separately made comments and recorded some information about case. The Stephen Wiley case was one that comes to mind. And, so they independently both recorded some things. And, I did not ever work with Gina Demas, but I did work with Joan Fons, and I think she's a pretty straight shooter. She was not one to mince words or to play around with things. I would trust her information. But that's just me personally. So, I mean, that's the best I can say.
Yeah, that's helpful. I mean, I think it's what you alluded to earlier, that these logbooks, there may have been the original data stored in the logbooks, but they don't necessarily exist anymore. So,
Right.
right, that's one other issue. And then the last question that I had, which I think you answered with your presentation, but on some of those worksheets that you showed, I noticed there was like a result on the left-hand column in one color ink and then a result on the right-hand column in a different color ink. And I was just wondering what that was, but I think you answered it in that they were for the two different tests. So one was
Right.
the test on the left and then on the right was the RH test. So is that correct? So I wondered if it was like a second reviewer, but that's not the case. It's just the two different tests.
Yes, and sometimes you would see the RH if it was limited sample size, for example. You might see that she would test just the big D antisera. She would use that one alone so that she could say RH positive versus RH negative and not do the whole panel. Yeah,
Okay,
you could report that.
thank you.
Mhm.
I think it's fortunate that we have someone with this information to help us answer some of these questions. So thank you very much. Anybody else of the subcommittee have any questions or thoughts that they want to bring up at this time?
I have two more to add.
Okay.
And again, having listened to some of what they were saying in the podcast, a part of this, they were talking about, when you go back and do an audit, and Director Jackson in her written response mentioned this, is that the lab is now fully accredited. That accreditation process is working in a sense. All you gotta do is look to your neighbor in the District of Columbia that's been shut down twice in the last 10 years, something like that, by the accreditation process. Houston Crime Lab, Boston Crime Lab. I mean, if you can see the process is working and doing these audits, and they're being done by outside people. So again, that's something, if I were talking to the reporters, I would raise that we're trying to clean things up. And that's what the accreditation process is doing in helping to standardize how the testing is done and what's acceptable and not acceptable. The other comment that about sort of the same thing is actually more that Amy will understand, is they changed the admissibility standard under the Daubert ruling out of the Supreme Court. And before that ruling, expert testimony was basically, if you were working in the field for 30 years, you could come in and say whatever you wanted to because you had the experience. Never mind that there was no science to support what you were saying. And that ruling, I think, has helped a lot in the legal community. Bite marks being a good example, fire residue examination, all ones that have gotten caught up in the Daubert, show me some science that supports what you're saying, Dr. Expert or Mr. Expert, and that I think has also been helpful. And again, it's something that could be pointed out, is that not only is the science evolving and its ability to police itself, the legal community has been putting in things that help them to police the labs as well. That's it.
Yeah, no, very, very well taken. I agree with both those points wholeheartedly. I also appreciated that we received the 2019 review of the previous serology review that was done, the DNA results. I was impressed by how comprehensive that was as well. So I think we have a lot of information. I think it's going to take a little bit for us all to go through all of these case files and information that we were given. So in terms of if the subcommittee doesn't have any other questions or thoughts at this time, I can open the floor up if there are any public comments. And just remind everyone that public comments are limited to 5 minutes. And I don't know if Director Jackson or Ms. Jenkins, if you can tell if there are any public comment, anyone that would like to speak?
Yeah, yes, we had 2 people that signed up to provide public comments. And the first is Ben Pavier from VPM, and I'm going to try and make him a panelist so that he will be able to speak and turn on his camera if he would like to. So, Mr. Pavier, hopefully,
Morning. Can you all see me?
yes.
Thank you all for taking this up. I'm a reporter at VPM who helped out some with the podcast. I'm not sort of the main driver of it, and so I'm probably less fluent in some of these ins and outs than other folks. But wanted to just chime in with a couple points. A couple things that were sort of left out of the synopsis. We had multiple sources tell us that Ms. Burton worked and conducted testing alone on evenings and weekends, again, alone. And also that Pete Marone, the former lab director, acknowledged that the lab couldn't get accredited while Ms. Burton was working in the lab, and it was only possible when she retired. And, you know, as I think you heard, there were multiple concerns brought to lab leadership about Ms. Burton's performance. And that when the DNA and testing notification project happened, the governor's office wasn't in the loop about the quality of Mary Jane Burton's work. The focus was basically on her having kept these shreds of evidence. So I know that Dr. Maha used the phrase that, you know, they did the best with what they can. I wonder if the department feels that way, if you all feel that like this, and agree with the commission in general agrees with the notion that a review here isn't possible, or that further action isn't impossible, is not feasible given what you've heard or learned from the podcast. And I know some of you maybe haven't had time to dive into it, so I'll just leave it there.
Thank you. I think I would say that at least for me, and I'm not speaking for anyone else, but at this time, I think honestly, there's so much information here that I think I would want to reserve any opinion until I've had a chance to review it all. I mean, certainly, I think the reason we're all here is because we would like to think it needs to be reviewed and then go from there.
Is there a sense of timeline at all on that? I mean, podcast has been out 6 months. I understand you haven't had some of the paperwork is quite as long.
Yeah, we, recently received the paperwork. I think after we have some public comment, we're going to probably try to set a next meeting date, and that'll kind of determine probably how quickly or it'll go. Any other comment from, if not, we can move on to the next commenter.
I was gonna have, this is George Maha again. Again, I can't speak for the department, but if you look at the accreditations that have come into play since the 1970s, they do have personnel standards. They do require management to be, effective. And so those are again, things that are forcing, the kind of things that were going on in the 1970s are not going on today. And in part, it's because the accreditation process is looking at if there's where there's problems, they're trying to put in appropriate standards to make sure that people are not making errors. So I would look at that. I'm more optimistic, I guess, in the sense that that I see the processes have come into place, and having been in this a long time, I can say that they have gotten much better and much stricter.
Or, yeah, I don't think, oh, sorry, I don't.
I say
guess
that's
I don't
also
know that-
true on the clinical side too.
I don't know that anyone on the podcast would, no one we talked to, I think, would dispute that. I think part of it is sort of, Mary Jane Burton touched a lot of cases, right? And there's a question, some of those people are, no longer alive, or the cases were thrown out, or, but some of them maybe did result in significant criminal penalties. So there's a question of how to handle that.
All right. Thank you very much. We appreciate the comment. Dr.
right.
Jackson, if anyone else?
Yes, let me, let's see. Sorry. Yes, we do have one other person. So Tessa Kramer who was the reporter on the podcast, had asked to give public comment, but she is not able to attend today. And Ellen Horn has messaged me and asked to speak in her stead, and I am working to make that happen.
Thank you.
Okay. Ms. Horn, you should be.
Hi there. I thank you all for being here and taking an interest in this. Obviously, Tessa and I'm the executive producer of the podcast. We have spent several years looking closely at this and interviewing lots of people who had direct knowledge of Miss Burton's work. There was a characterization of our podcast and our reporting as not providing information and being somewhat withholding. And I just wanted to really make clear that we're available and interested in sharing what we know. Simply the timeline that you laid out for when we gave information, we wanted to finish our publication and finish our fact checking and review process before we published. We have an obligation to confirm the truth and to do the best of our ability to do that. Our public media partners at VPM have been incredibly helpful in helping us to get to publication. But at this point, we are really interested in finding out what DFS is going to do and what you guys are going to do, really to take responsibility for what we've learned. So if we can help in any way, we're there and happy to do so. Thanks so much.
Thank you. We appreciate that comment as well. Director Jackson, do you know if there's anyone else public comment?
I do not. I know that there's one person. Hold on 1 second. Let me. I know there's one person who is an attendee that is on the phone and would not be able to raise their hand. I just want to check in with that person. But I do not see anyone else. Thank you.
Okay. Thank you. I just want to give the members of the subcommittee another opportunity. I know there's a lot of information today. Is there? Oh, Dr. Vallone looks like he has a question.
I think it was just more for us or for you to kind of like, maybe we can at some point spell out next steps. Like you said, there's a lot of information and just how do we want to address it and kind of what are the goals for addressing it, to be more specific. If there's a lot of information and a lot of information in the podcast as well. So I don't know if that's something we discuss now or at a future meeting.
Yeah, thank you for that question. I think it's a good one as well. I guess my thought on it, and I'm welcome to others' opinions, is that the way I kind of work, the way my brain works is I feel like I have to look at everything before I even can decide, what the next steps are or what the questions might be. What I thought I was thinking I would be doing would be looking over all the information. So we have the information from the podcast, and as Ms. Jenkins said we can, that's all in transcript, so we can read that if we need to refresh on that. We have the information provided us from DFS that came from VPM. We have the case files from DFS. So I was thinking, perhaps just reviewing that information and then reconvening, to see if we had, if there's issues that come up that we think need further investigation, or if there's other information that we might need from DFS. So that's, it's not clear to me what our next steps would be until we actually read through it. I mean, perhaps, unless I would open it up to Director Jackson and Ms. Jenkins, if there's if they have any other ideas on perhaps an agenda or questions that we could have the subcommittee particularly look at, or anyone else on the subcommittee.
So I do think there's some additional information that needs to be provided. I had added in the last week or so, as you pointed out, Dr. Corrado, the DNA project overview from the Crime Commission, and the serology review overview as well. There's, I think, on those 5 cases, there's additional information I can provide in addition to the case file as far as outcomes and what occurred. And there's also, I provided a link on a US DOJ desk book on serology from 1971. I'm not indicating that that was on Mary Jane's desk. I do not know the answer to that. How I was able to get to that desk book was an article by John Morgan dealing with a review of these wrongful convictions that you all might be familiar with, that came out in 2023. And that was a reference cited in that paper. And that reference speaks in terms of bacterial contamination. I have not provided you with the serology exoneration documentation. I don't know whether I felt like I was burying you all in paperwork, and I didn't want to do that. So whatever you all feel you might need additionally, we are helpful. We are ready to provide whatever you might want us to provide.
Thank you. I guess maybe one thing, and I guess I'll ask the other subcommittee members to weigh in on this as well. It might be helpful if you could prepare for us a specific list of what, I'll say, the allegations are, if that's the right term or not, that, the podcast has brought forward. Maybe a specific list of those things, so that those are things that we can have, be thinking about when we're reviewing the documents. I mean, I think we've all sort of heard it, but it's a little bit hard to keep track of it all, and if we're all doing it differently, it might be good if we all have one list to go off of.
Yeah, that would be fine. I'd be happy to do that.
Thank you. Any other subcommittee members, do you have any other suggestions of anything else we need to do? And I guess if we do determine a date for the next meeting, I'm sure that we all would like to make sure we do this as in as quick of a fashion as possible. However, I know that we're coming up on November, December, which are always quite busy and in less time that people have. So thinking about the next meeting date, so think about the amount of information we have to go through and what your schedules might be like in the next few months. We could try to schedule something in November, December. My feeling is, I think it might be best to wait until perhaps the beginning of January, perhaps the week of January 8th, so we get past the beginning New Year's. But if other subcommittee members, if you think that, second week of January would work well, or does anyone think we need to meet prior to?
Hi Kathy, it's Jen. I think January works best, especially if we're gonna have like those, a more detailed list of kind of, I think, spelling out and helping a little bit of kind of what we're looking for through the documentation. But I do think really to go through it, it's gonna be a fairly extensive look. I think it takes a fair amount of time, at least from what I've done so far, which I don't think was really even close. So, and I felt like I got through a lot of stuff, but I still think we have to go back. Just, I think based on what Pete was saying too, it's not just having a more, concise, what are we looking for exactly? I mean, we know what we're looking for in some respects overall, but when we have some of these questions or allegations spelled out, I just think it's going to be a lot more helpful when we take this dive in there. So.
Yeah, thank you. I agree with that. So it'll take a little while for the department to get us that list, and then again for us to go through all these things. I would propose then that unless anyone has any other comments, that we perhaps try to schedule a meeting for the I'm sorry, I'm just looking at my calendar now. I guess it would be the second week of January, so the week of January 8th. Also, just so everyone understands, this meeting, due to the regulations, the Virginia regulations, it will have to be an in-person meeting because I think we have to do every other, one can be online and other meetings have to be in person. So the subcommittee the members would need to meet in person, assuming it would be at the DFS headquarters in Richmond. So just take that into account. Of course, I'm sure they'll help us with travel arrangements, but some of us, myself at least, have to fly. So I know we'll have to make those arrangements soon to make sure we can get them in time. Is there any particular day of the week of that week? Well, first of all, does that week work does anyone have a conflict with that week? Let me say it that way. No, I don't hear any, see any. Is there any particular travel wise or people's work wise, any particular day of the week that does not work for them?
Kathy, it's Jen. Just Monday, it's my son's birthday. So, but I can make any other day that week. I would just like to be home Monday if I can.
Okay, that's fair. Because I will be flying, I suppose I will probably need to come in the day before, and I don't know, I think perhaps Erin, you would be flying as well.
Yes.
Assuming that. So we could make the meeting for that Tuesday or Wednesday. If that works okay. Does anyone have a preference, Tuesday or Wednesday? Anything with DFS, I mean, I don't know, do you have other things scheduled for your office at that time that would prohibit us from meeting there? No?
Not that
No.
I'm aware of.
When is the forensic science board meeting? It's a week before.
It's the week before on the 3rd.
Right. The General Assembly in Virginia convenes on that Wednesday, but there shouldn't be any reason that you couldn't meet any day that you would wish.
But
if Tuesday works, that might be nice.
well, so that's my question. I guess,
Yeah.
would you prefer, would it be easier for your staff, knowing you might, I don't know if you have to travel for that meeting or not, for us to meet later in the week, or does that Tuesday work? Or we could meet Thursday if that's, I don't know what you all have to do for those meetings. I'm sorry, Linda, or I didn't hear that.
I was just going to say it generally at the beginning of the General Assembly session, it's that first day, generally there's nothing that we would need to do. necessarily. As it progresses, then there are meetings that we may have to provide testimony to a subcommittee or something like that. But right at the beginning, generally, it should be okay. So either that Tuesday or Wednesday really should be fine.
Okay, well, I would suggest then that we meet on that Tuesday, the 9th. That'll give us a chance to come in the day before if necessary, and hopefully we'll work so you're not tied up for these other meetings. Is everyone okay with us? I don't think we need to, do we need to do, Ms. Jenkins, do we need to do a motion for this, or can we just set the date?
I think you're fine with just setting the date. I don't think you should set a motion as long as everybody's in agreement.
All right, everyone is agreement with meeting on January 9th, Tuesday.
Yes.
All those, yes, okay, great. All right, I think at this point, then that concludes our agenda. Oh yes, Mr. or Dr. Valone, sorry.
I said I have one more question, so we'll get some further information from DFS on kind of things we should focus on. And I was just asking, is it okay or appropriate if we had any I guess informal meetings to discuss between ourselves before the 9th, or should it all be public on the 9th?
Must be public on the 9th.
Okay, that's fine. Thanks.
If you have specific questions you want to email to me, that's fine. But there can't be any meetings. We should all be together and discussing things on the 9th. Okay.
That's great for clarity. Thank you.
Thank you for the question. Anybody else before before I ask for a motion to adjourn the meeting? Yes, Erin.
I just a thought, there have been reviews of these case files previously, right? In the post-conviction testing, is there any document about the audit plan or the scope or what people were guided to look for when they did that audit documented anywhere? I'm just wondering kind of what the method was, I guess, in that audit.
Yes, does someone from DFS, could you answer that question for us?
Mr. Jenkins, are you going to answer that question? So go ahead.
I can provide some information. So as far as the audit process, if you're referring to the post-conviction testing, there were basically three criteria. Was evidence taped down in the files? Was there a conviction? And what was the crime type? And so the homicides and sexual assault cases, those were picked out for DNA testing if it met those criteria. There were serology cases that were also done on other types of cases. And a number of the cases, you may not have conviction information. And so that was, and it was just a review, a paper review, not a computer review, because these are not in computers, but file by file, looking at the files to identify if they have taped down evidence.
A separate issue was the Washington case, and that case was the subject of audits, both internal and external, as well as Judge Humphrey's review team of issues pertaining to Washington as well. I think Dr. Maha may have been on that review team if my memory serves correctly. And so I can provide all of those audit documents as well.
Thank you. You did provide, we do have a report from the PC testing though, correct?
Correct. I had not provided the audits regarding Washington, so I can provide those documents as well.
Okay. Ms. Forey, does that answer your question, or was there something further you're thinking of?
Thank you.
Yep. Great. Any other questions? Oh, well, I want to thank everyone again for their time, and I would ask for a motion for a meeting to adjourn.
Motion to adjourn?
Any
Second,
seconds?
second.
Thank you, Dr. Maha. All those in favor, say aye.
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 Any opposed? Hearing none, the motion passes and the meeting is adjourned. Thank you all very much. We'll be in touch.
Thank you.
Thank you.
Thanks.